IB Bio, Option B, Biotechnology, Bioinformatics Part 7, is the first of two movies that look at B.4, Biotechnology in Medicine. Part 7, B.4a, this movie, will have its focus on the use of biotechnology in the diagnosis of disease. The essential idea is biotechnology can be used in the diagnosis and treatment of disease. Here is the outline of all the movies available for Option B. Use this outline to find the movie you need for review. This movie is focused here, but I'd like to comment about the B.4 movies. The first one covers the biotechnology, the use of biotechnology in diagnosing disease. The second one examines diagnostic techniques related directly to DNA, as well as the use of biotechnology in providing treatment. So let's jump right in with the first IB syllabus statement. Metabolites that indicate disease can be detected in blood or urine. And just to ground our understanding, let's define the term metabolite. Metabolites are the intermediate molecules and products of metabolism. You should be familiar with metabolites from your study of cell respiration. Pyruvic acid, lactic acid, acetyl CoA, these are all metabolites. NADH, FADH2, ATP, all metabolites. Fatty acids, glycerol, amino acids, these are all metabolites. The presence of an excess or low level of metabolite would be evidence of a non-functioning metabolism. The medical community examines the level of metabolites in blood and urine to diagnose disease. Uric acid is a metabolite found in the blood when nucleic acid metabolism is not functioning properly. In Lesch-Nyhan syndrome, the metabolism of purines, adenine and guanine, is not functioning, resulting in uric acid crystals that can be detected in the urine. In this image, we can see uric acid crystals in the bloodstream. The metabolite homogentisic acid can be detected in the blood and in the urine when someone suffers from the disease known as alkaptonuria. Alkaptonuria is a genetic disease caused by the inheritance of two recessive alleles. People with alkaptonuria have a malfunction in the metabolism of amino acids, phenylalanine and tyrosine specifically. The inability to metabolize phenylalanine and tyrosine results in the accumulation of homogentisic acid, which in its oxidized form produces a dark color. In these images, you can see the accumulation of homogentisic acid in the connective tissues, evidence of its presence in the blood and in the urine. Normal urine on the left, upper left, and the urine of someone suffering from alkaptonuria on the right. The accumulating homogentisic acid causes damage to cartilage specifically, leading to arthritis as well as problems with heart valves. Glucose is a metabolite found in the blood. Tests that detect excess glucose in the blood or the urine provide evidence of disease such as diabetes. PKU is another inherited disease related to amino acid metabolism. The homozygous recessive condition results in a non-functioning enzyme, phenylalanine hydroxylase. Phenylalanine is not transformed into tyrosine, but instead is metabolized into phenylpyruvate that accumulates in the blood and the urine. Untreated PKU can lead to intellectual disability, seizures, and mental disorder. Cholesterol, high-density lipoproteins, low-density lipoproteins are metabolites whose concentrations in the blood can be correlated with coronary heart disease. The concentrations of these metabolites can be measured in the blood. If the concentration of cholesterol, high-density lipoprotein, low-density lipoprotein, or triglycerides as metabolites is above a certain threshold, doctors are concerned about the possibility of heart disease. There is a correlation between the levels of cholesterol and high-density lipoproteins and the development of plaque in the arteries that could lead to a heart attack. The detection of blood metabolites to diagnose disease is done routinely, rapidly, and with accuracy. Timely diagnosis leads to early treatment. Early treatment prevents long-term complications for both genetic disease and infectious disease. In the case of infectious disease, diagnosis involves isolating the pathogen by taking swabs or stool samples and culturing them, as can be seen here. A pathogen is a disease-causing organism. The detection of a blood parasite, such as these trypanosomes that cause sleeping sickness, can be more complicated, but diagnosing sleeping sickness is certainly possible. Biotechnology, as a development in scientific research, now provides new ways to diagnose and treat disease. So that brings us to this IB syllabus statement. Infection by a pathogen can be detected by the presence of its genetic material or by its antigens. And just to ground our understanding, let's define the term antigen and provide a bit of background on pathogens and antigens. An antigen is any molecule recognized as foreign that stimulates antibody production. Let me provide some emphasis here. An antigen is any molecule recognized as foreign, and that molecule could be just a small part of the larger pathogen. A surface protein is an antigen. Antibodies are secreted by your immune cells, B cells specifically, and antibodies are secreted against the antigens. We'll see more on that later. Antibodies target the molecules, they target the antigens on the surfaces of pathogens. Now, can you distinguish the terms antigen and pathogen? An antigen would be any molecule as part of a larger pathogen that stimulates antibody production. In this diagram, you can see the antigens, which are foreign molecules as part of larger pathogens, and you can see the antibodies, these Y-shaped structures. Antibodies are proteins secreted into the blood by B cells of the immune system in response to the antigen. Antibodies bind to the antigen, neutralizing the antigen by clumping the antigen, a process called agglutination. In this diagram, we have bacterial pathogens and viral pathogens, each with antigens on their surface. You can see antibodies that have been secreted by the B cell that will bind to the antigen and neutralize the pathogen. So the presence of antigens and/or antibodies in the blood can be used to diagnose disease, diagnose the presence of the pathogen. So let's imagine with this diagram that the blood, seen here in the bloodstream, has the hepatitis C virus with its surface antigens and the antibodies that have been secreted against the hepatitis C virus. The presence of the antigens and the antibodies in the blood can be used to diagnose disease by withdrawing blood and using various techniques to detect the antigen or the antibody. Stay tuned. Monoclonal antibodies are used as a rapid method to diagnose hepatitis C infection. Monoclonal antibodies can be designed to bind either the antigen on the surface of a virus, such as Hep C, or bind to the antibodies secreted by the body against Hep C. Monoclonal antibodies can be used to detect the presence of the infection. Viral pathogens infect cells, thus their concentration in the blood may be quite low and too low to measure, as is the case with HIV infection. Thus the presence of antibodies in the blood, detected by the use of monoclonal antibodies, allows us to diagnose HIV infection. Again, we detect the presence of antibodies in the blood, not necessarily the virus itself. And now, I will not cover the design or specific use of monoclonal antibodies in this movie or as a part of option B, but I will cover a similar diagnostic procedure called ELISA. Stay tuned. So that brings us to these IB syllabus statements. Describe an ELISA test. Interpret the results of an ELISA diagnostic test. ELISA is enzyme-linked immunosorbent assay. It is a technique used to detect proteins, antigens, or antibodies in the blood that determine if infection from a pathogen has occurred. A pathogen's antigens stimulate the production of antibodies in the bloodstream by B cells. ELISA is used to determine infection by detecting the presence of the antibody or the antigen. All right, now in moving forward, just to mitigate possible confusion, you will see antibodies used in different ways. Pay close attention. In the ELISA technique, antibodies are used in two ways as tools of the technique. And we can't forget that there are antibodies in the blood serum of the patient that we mean to detect. So here is the technique, and I'll give you a number of images over the next few slides to help you understand the ELISA technique so that later on you can interpret a result. An antibody, seen here in green, an antibody to the antigen is fixed to the solid surface. This might be a monoclonal antibody. Now patient serum, blood serum, is added. And if the antigen is present, in other words, if the patient is infected, the antigen binds to the antibody. Now, just to be clear, the antigen in the patient blood serum could instead be an antibody to the antigen. In this case, we're dealing with the antigen itself. Just keep this difference in mind. Now, add an antibody that's attached to an enzyme. This is called a labeled antibody, and it binds to the antigen itself. These two antibodies are tools in the technique. Both bind to the antigen in the blood serum. And then we add a substrate to the enzyme. Color change occurs due to the enzyme, and the quantity of color on the plate is proportional to the concentration of the antigen, the presence of the disease. I'll be showing you more diagrams to help you understand the ELISA technique, but here is the technique in words. You can stop the movie now to take notes or follow along with me. If the blood is being tested for the antigen directly, as it was in my first example, antibodies against the antigen, seen here in blue, are fixed to a surface. I'm referring to these antibodies as monoclonal antibodies. These are part of the ELISA toolkit. Blood serum is added, and if the antigen, seen here as orange, is present, they bind to the fixed antibody. Then a labeled antibody is added, seen as green here. The labeled antibody has an enzyme, and it also binds to the antigen. The labeled antibody with its enzyme or a fluorescent molecule allows the technician to view a positive test when a substrate is added. If the blood is being tested for the antibody to the antigen, this is called an indirect ELISA test. Instead of a monoclonal antibody, the antigen itself, seen here as blue, is fixed to a surface. Blood serum is added, and if antibodies are present in the blood, seen here as orange, they bind to the fixed antigen. Then a labeled antibody is added, seen here as green. The labeled antibody binds to the blood serum antibody. The labeled antibody has an enzyme or fluorescent molecule attached that allows technicians to view a positive test. One more time, but this series of slides adds an important piece of information. The fixed antibodies are located within wells. The plate has numerous wells for numerous blood samples from different people. So here is the fixed antibody on the surface of a spot or a well. Blood serum is added, and if the antigen is present in the blood of the patient, it binds to the fixed antibody. Then a detection or labeled antibody is added. The detection antibody has an enzyme or a fluorescent molecule attached. Then add a substrate that changes under the influence of the enzyme. Fluorescence, light, or color would be produced. And if the well changes color, as you can see here, we have a positive ELISA test. The serum of this individual has the antigen or the antibody to the antigen. The person from whom this blood sample was taken has been infected with the pathogen, viral or bacterial. The greater the color change in certain wells relative to other wells, the greater the concentration of the antigen. In other words, the more developed the infection is by the pathogen. The advantage of using ELISA is primarily the speed with which results are obtained, roughly 2.5 hours. However, the cost is high due to the technical nature of the procedure, including trained staff. ELISA can be performed to evaluate the presence of antigen or the presence of antibody in a blood sample. It is a useful tool for detecting viral infection because viral pathogens by their nature get inside cells out of the bloodstream, out of sight. Thus, ELISA is a useful tool for determining serum antibody concentrations as a test for HIV infection or, for example, West Nile virus infection. As well, ELISA has applications in the food industry, detecting potential food allergens, such as from peanuts and eggs. ELISA can also be used in toxicology as a rapid screen for certain classes of drugs. Now that brings us to the end of ELISA. Let's move on to another diagnostic technique. And here are the relevant IB syllabus statements, our last of this movie. Describe the use of tracers in tracking experiments that provide information about the localization and interaction of a desired protein. State that transferrin linked to luminescent probes is used to track tumor cells. Transferrin is a glycoprotein with 2 atoms of iron and an added fluorescent or radioactive label. The PET scan, seen in the right image, tracks the location of transferrin in the body. The procedure goes as follows. A small amount of transferrin is given via a vein injection. The patient waits for an hour or so as the tracer, the transferrin, travels within the body. Transferrin moves through the bloodstream and accumulates in organs and tissues that use transferrin, particularly the iron. The patient then lies on a narrow table that slides into the large tunnel-shaped PET scanner, PET scanner. It takes about 30 minutes to complete the procedure. A positron emission tomography, PET machine, detects the energy emitted by positively charged particles called positrons that are emitted by the radioactive or fluorescent label. The image displays the concentration of positrons according to brightness and color. Transferrin is a glycoprotein with 2 atoms of iron that binds to a specific receptor on the surface of vertebrate cells, and it delivers up to 2 iron ions via receptor-mediated endocytosis. Transferrin is fluorescently labeled, thus it can be visualized as it attaches to cell membranes or as it enters cells. In these 2 micrographs, we can see transferrin fluorescing as it attaches to and enters cells. The left image, no transferrin. The right image, we can see the presence of transferrin. Left image, no transferrin. But in the right image, we see this cell that has picked up transferrin. Now, tumor cells display high levels of iron uptake relative to normal cells. Thus, transferrin is seen in higher quantities in tumor cells. In this diagram, we have a PET scan of the pelvic region of a mouse following the injection with transferrin. The arrow points to a prostate tumor. The red regions indicate high transferrin uptake. Now, tumor cells seen here overexpress transferrin receptors and CD44 receptors. Now, Fe3O4 magnetite with hyaluronic acid binds to CD44 receptors. Transferrin binds to transferrin receptors. Now, when the Fe3O4 with hyaluronic acid is linked to transferrin, as we can see here, following their injection and a PET scan, they provide strong evidence in the diagnosis of a cancerous cell. Now, just as a last note and a side note on the transferrin story, poisons are being attached to transferrin, in this case rubidium, probably radioactive. Transferrin is delivering the poison to cancer cells, causing cancerous cells to die. The poison is taken up and causes cancer cell death. This is a way of using transferrin to target cells and kill those cells. Now, as we come down the home stretch of the movie, it's important to remember that tracers other than transferrin are used, and I'd like to provide some examples. Here is a PET scan of the brain following the injection with radioactive fluorine. The arrow points to a brain tumor. 18F-FLT accumulates in proliferating cells, cancerous cells. 18F-FLT accumulation indicates the activity of the enzyme thymidine kinase, a protein that characterizes high levels of cell division, which are typical of a cancerous cell. Here is another example where we use radioactive fluorine to diagnose cancer. The radioactive fluorine is attached to a larger molecule known as COX-2. COX-2 is injected into the body and targets an enzyme active in tumors. COX-2 targets a protein, an enzyme, in tumors, and the radioactive fluorine is seen in the PET scan, providing a diagnosis of cancer. Now, this is a protein tracking experiment that provides a diagnosis of cancer. Now, various radioactive isotopes are used in medicine. The radioactive atom is tagged to a natural chemical like water, like glucose, methionine, ammonia, and the combination is called a radiotracer. These radiotracers are used in protein tracking experiments that allow us to diagnose disease. Over the last dozen slides or so, I've been working on this IB syllabus statement. Describe the use of tracers in tracking experiments that provide information about the localization and interaction of a desired protein. And you need to be able to state that transferrin linked to luminescent probes is used to track tumor cells. BioLume Incorporated has exclusive licensed rights to more than 40 U.S. and foreign patents covering natural bioluminescent proteins. The bioluminescent proteins are injected into a patient and provide images where light is produced by a chemical reaction within a living organism. Unlike fluorescence or radioactivity, bioluminescence does not require energy from another source of light to create the illumination, and bioluminescence is not radioactive. Bioluminescent proteins are derived from deep-sea marine organisms that create their own light. Bioluminescent imaging allows for early diagnosis of disease even before there are symptoms, and bioluminescent proteins can detect tumors. In the images on this slide, we can see vascular imaging in the upper image and bioluminescence in a mouse and in a brain. This is the future of biotechnology in diagnosing disease. And that brings us to the end of IB Bio Option B, Biotechnology, Bioinformatics, Part 7.